Hello, my name is Felix Upon. I'm a research assistant in Dr. Atkins' Fly Lab, and I'm glad to be presenting here at the URS 2021. So what I'm working on in lab is a strategy to build a stock in Drosophila Melanogaster to study ATPsyn-based subunit gamma function in larvae. So to set the stage for what we're going to be talking about today, it's important to understand the hippo pathway. So in Drosophila melanogaster, the hippo pathway regulates organ size development in Drosophila. So the hippo pathway is composed of kinase, and essentially a kinase is an enzyme that puts phosphate group on other molecules. So the hippo kinase phosphorylates warts, then warts phosphorylates Yorkie. However, the phosphorylation of Yorkie itself inactivates Yorkie. So when Yorkie is inactivated, then organs grow normal sizes in flies. So our lab has identified that the hippo pathway shows some genetic regulation by bellwether, and bellwether is the alpha subunit of the ATP synthase machinery. So we wanted to see if this interaction is due to a specific bellwether function or to just the ATP synthase machinery. So to test this, we're manipulating another subunit of the ATP synthase machinery, which is the ATP synthase gamma subunit. So this manipulation is done by expressing RNAi against the ATP synthase gamma subunit, and then examining the effects of the ATP synthase gamma subunit in growing larval tissues and compare it to bellwether and its effects in growing larval tissues as well. However, the ATP synthase gamma RNAi is balanced over TM3 chromosomal balancer. So for the flies that have the TM3 chromosomal balancer, along with the ATP synthase gamma RNAi, essentially are undetectable in larval stage. We can't really see what we're looking for in the ATP synthase gamma RNAi on a fly with TM3 chromosomal balancer when it's in the larval stage. So then ATP synthase gamma RNAi must be rebalanced over TM6B, because TM6B is a chromosomal balancer that is readily detectable in larval stages. So that's the idea of the presentation today, to present the approach as to how we go about rebalancing ATP synthase gamma RNAi from TM3 chromosomal balancer onto a TM6B chromosomal balancer. So in lab, we work with Drosophila melanogaster a lot. So Drosophila melanogaster is really convenient for our lab because they reproduce very quickly, about 10 to 12 days. And to walk through the life cycle, as a result of mating, an embryo would be made, and about a day or so after the embryo, it enters the first larval stage, and about a day or so, the second larval stage, and about a day or so, the third larval stage. For our presentation today, we'll be focusing in on the third larval stage, and about 2 or 3 days after the third larval stage, the pupa arrives, and after pupa, about 3 to 4 days after, the mature fly arrives. So it's very quick, it's fairly straightforward, so we know what to expect and we know what to look for on each stages of the fly. So moving on to our methodology and how we approach this problem, we understand that flies that we work with have 4 chromosomes, and today, in terms of ATP gamma RNAi, we're focusing in on the 3rd chromosome. So to be able to know where the ATP synthase gamma RNAi is located and how it's working on the chromosome it is on, we need balancer chromosome. So balancer chromosomes have dominant markers, so then we'll be able to see them physically as they are expressed on whatever chromosomes they are on. But the problem is balancer chromosomes have recessive deleterious mutations. So it is important to have a strategy to preserve these mutations so that our balancer chromosomes don't die off. So ATP synthase gamma RNAi is homozygous lethal. So having both recessive alleles of ATP synthase gamma RNAi on a chromosome would essentially die. So that gene would essentially die off in the genetic population. So to counter that, what we do is we complement a balancer along with a mutant gene, so that where the mutant gene is defective, the balancer will complement that part, and vice versa. Mutant genes are heterozygous, so then a balancer that is recessive, when coupled with a heterozygous dominant mutant gene, the balancer essentially acts as a carrier, and then we are ensured that the balancer is still within the population, because double alleles of homozygous lethals is what we're trying to avoid, because that's how we lose our balancer chromosomes and its effect in our genetic population. Another important feature about balancer chromosomes is that they have chromosomal rearrangements, and that's also another important way for us to recover from recombinants. So we need to keep our balancer chromosomes the way they are. We need to keep them essentially healthy and alive in our populations. So with the work of inversion breakpoints, we essentially inhibit synapses so that crossovers don't happen near the vicinity of the inversion breakpoints. And if crossovers that happen within the inversion breakpoints essentially wouldn't be aneuploid, so then the progeny of aneuploid are not viable, so we don't have to worry about those because they can't really reproduce and then make progeny that does not contain our balancer chromosomes. So that's clever ways that we have to ensure that our balancer chromosomes are still active in our genetic population, because our balancer chromosomes do have dominant markers, and those dominant markers will allow us to see the genetic information that we're looking for based on the phenotype of a fly or larva per se. So the ATP synthase gamma RNAi, like the name suggests, has an RNA. So RNA in general causes destruction of target mRNA, so thereby seizing the processes or the mechanisms that would continue on if mRNA was to reach its destination. And this then would decrease or eliminate the amount of proteins made, and loosely we term it knockdown. So moving on for today, we're mainly talking about two chromosomal balancers, and both these chromosomal balancers are dominant markers. So we do see them phenotypically in flies and one in larvae. So for TM6B, as a chromosomal dominant marker, its phenotypes is long bristle hairs on the back of the fly, along with humeral, which is essentially at least 3 hairs on the shoulder of the fly. For TM3 chromosomal balancer, its phenotype would be short or stubble bristle hairs on the back of the flies. And in the larva, the TM3 chromosomal balancer looks a lot similar like a cat nest. And why this is important is because cat nest larva is a normal larva. Essentially, it's a wild type. It doesn't really have the genetic manipulations that TM3 chromosomal balancer has. It's what we compare other genetic modifications to. So here, in the adult, it is different because the hairs on the back of a wild type fly cat nest or a normal fly cat nest is very different from that of a TM3 chromosomal balancer. So in adult flies, it's fairly easy to tell, so there is no problem here. But in larval flies, a TM3 chromosomal balancer doesn't have enough distinctive phenotypes for us to tell it apart from a wild-type larva. So that's the problem we have. We don't know which is which, and we run the risk of dissecting wild-type larva in search of ATP synthase gamma RNAi balance over TM3. So to combat this ambiguity, we just rebalance ATP synthase gamma RNAi over TM6B chromosomal balancer, because TM6B chromosomal balancer has a distinct shape and size in the larva enough for us to tell it apart from a normal wild-type larva. So we drastically decrease our chance of making mistakes in dissecting the wrong larva. So Mendelian genetics offer a way to explain how we go about rebalancing from TM3 onto TM6B. So in the third chromosome, homologous chromosomes, the ATP synthase RNAi will be on, along with the TM3 balancer on the third chromosome. So in meiosis, they pair, then they separate according to Mendelian segregation. But because TM3 chromosomal balancer doesn't have enough phenotype in its larva, it looks a lot like a normal larva, even though it may still have the ATP synthase RNAi on it. We can tell just by looking at it the difference between a TM3 chromosomal balancer from a normal larva. So the strategy here is to rebalance it over TM6B. And so again, on the same third chromosome, carrying the ATP synthase RNAi, but this time we cross it with a fly that has a TM6B chromosomal balancer. They undergo meiosis, homologous chromosomes pair, they separate according to Mendelian genetics. But this time, because TM6B has enough phenotype in the larva, we can tell it apart from a normal wild-type larva. And we can know that it has the ATP synthase RNAi balance over it due to the combinations of the chromosomes through meiosis. So then we know that this is ATP synthase RNAi over TM6B because of the phenotype, that's TEBI, because of the size. And we also know this is also ATP synthase RNAi over TM6B because those were the original parent flies that we crossed them with. So then we are sure that this has ATP synthase RNAi balance over TM6B, and so is this. So this diagram here offers a practical way as to how we do rebalance gamma ATP synthase gamma RNAi from TM3 onto TM6B. So we start off with our parent fly. So we know that our parent fly with ATP synthase gamma RNAi is balanced with TM3, but then the problem is we can't see it. We can't tell it apart from a normal larva. So we cross ATP synthase gamma RNAi that's balanced with TM3 with TM3 that's balanced with TM6B. And the idea here is to select against TM3. So we select against TM3 by selecting for characteristics or chromosome or phenotypes to look for, in the TM6B larva. So we select for TEBI and humoral because TEBI is a phenotype of the TM6B and humoral is also a phenotype of the TM6B in the adult flies. So we select for those, then we cross them back in the first filial generation in order to accommodate for the Y chromosome that was on here, that's gamma 1. So we cross them back. Again, we select against TM3 by selecting for characteristics on the TM6B chromosomal balancer, which is TEBI and humoral. And then in the second filial generations, we can be assured that ATP synthase gamma RNAi is balanced with team 6B. And how we know this is because Mendelian law of segregation ensures that the combinations and the pairing of the homologous chromosomes during meiosis would yield ATP synthase RNAi over TM6B, and our phenotypes is a way for us to know that we're looking in the right progeny. So then we can then use gamma to study bellwether, along with bellwether, to study how they both function in the Hippo pathway. So with gamma balance over the right chromosomal balance, which is TM6B, we can dissect for the right larva. We can look at the cells of both gamma 1 as well as bellwether, and we can see how we can see if bellwether truly has its own function as it relates to the Hippo pathway, or the ATP syntax machinery has the main function as it relates to the Hippo pathway based on the comparison of gamma 1 and bellwether. And with that, I would like to acknowledge my lab mate Harris Bioma and Dr. Atkins for giving me the chance to work in her lab and for helping me do this project. And with that, thank you to my audience and have a good day.